congratulations with the funding of your project. I also heard you were busy on a VENI now.
Yes.
And so also an ME, and that would be, if that's successful, that would be a major achievement for the field, I think. So let's hope for the best. But first, see your presentation.
Thank you so much. Is it here? Yeah. So thank you so much for the.
Can you try without the mic, because some people online still have an echo. So try to without the mic. I'll ask them if they can still.
What about now? Well, for those that doesn't know me, my name is Felipe. I'm postdoc under supervision of Inga. She was kind enough to let me present. I'm hands-on on both projects that are in the schedule right now. They're interconnected somehow, and basically we established the brain bank.
Okay, no, take the mic.
Take the mic.
Hey,
Yeah. Maybe if I go a little bit lower like this, is it good? Well, so basically the idea it is that we would establish the first brain bank, brain donor program focused on the harvesting of post-mortem tissue for biomedical research in ME/CFS. We have now two projects focused. The first one that is focused on neuro-endocrine mechanisms and neuroimmune mechanisms. The second one we could unfold from histopathological characterization to more genome-wide transcriptomic techniques as well. So basically I lumped up two presentations and then we can have questions afterwards if that's convenient. I think that's the most straightforward way. So the first project is titled Brain Changes in ME/CFS, focused on the stress and immune systems. As I said, we have two branches. Basically was the establishment of the brain donor program using the infrastructure of the Netherlands Brain Bank and all the expertise there. And the second one was the research arm that had three work plans: brain stress control mechanisms with HPA axis, brain immune alterations, mostly focused on microglia and T-cell infiltration, and a third research project that is mitochondria related. I'm going to give a brief update on these and these. The third work plan, it's for the next one, I'm afraid. So basically, the idea of a brain donor program is that we have people interested in contribute to research, and by doing that, they donate their brain after their death. The Netherlands Brain Bank has extensive expertise in this. We have now over 5,000 people registered, and we conduct over 5,000 autopsies like that already. And the ME/CFS brain donor program started in April of 2024. We already have 61 live registrations, and the aim is 200 by the end of this project. And so far, we did 10 autopsies, and the aim it is 50 by the end of 10 years. That's a little bit of the metrics. About the communication and dissemination activities, we have extensive contact with TEL and ME/CVS Netherlands, which we discuss the donor recruitment. I have the folder that Inga showed. If you have interest or if you'd like to know more, please let me know. But more than that, because of the implementation of the second grant, Lou Cursus also will integrate our team. We're still fine-tuning the periodicity of our meetings, probably monthly or bi-monthly, still like that. Also, in the efforts of integrating the target groups, we have a discussion with physicians. Ruud Vermeulen, sorry, his name is very difficult for me, from Utrecht, and also Hanneke van Santen. She provided inputs and is going to provide more inputs because she's a specialist in hypothalamus. As I said, there is ME/CFS flyers from the NBB where we basically present the whole pipeline, all the concerns about brain donation, who should be informed, what are the implications, and so on and so forth. We also have our own newsletter that has a space dedicated for ME/CFS research, as other research that is funded by or integrated in the NBB. We also have contact with Cardiozorg because they know that some patients of theirs have the interest in donating. So we are now talking with them, introducing the idea of brain donations for those patients. Of course, this should be done with care and prudence. So that's something that we are working on. And basically, we also provide tissue dissemination. So Rob is the first one actually that outside of our own group that has tissue from the brain bank to try to understand it in the framework of ME/CFS. He is trying to detect lipofuscin in brain tissue in collaboration with us. And because of efforts in conferences, there are also interest from abroad. So people in Australia, people in the US are very interested in applying for tissue. Well, recently the Netherlands Brain Bank had its anniversary, was 40 years. We had the honor to receive the Queen, and she could visit our infrastructure, see how post-mortem brain research is established, what we do, what are the outcomes and implications. Was an event that is not tailored to ME/CFS, but I heard from Inga that the Queen herself asked about long COVID and ME/CFS efforts, unprompted. So that is very nice, and that actually the whole event and the presence of her brought a lot of attention to brain banking and the societal importance of that. So we are very happy and pleased with this. More than that, in terms of dissemination, I could go to Australia, invitation by Sonia Marshall. They were very impressed with the quality of the NMCB related research, the efforts reached down there, and they also were kind enough to grant me a Rising Star Award because they recognized the potential of the transformative potential of the brain postmortem research. I could even compete with the scenery, the Australian scenery, so that was very nice. Well, the first work plan, the people that were already working in the first block already know. So basically, we were doing the HPA axis. Basically, the HPA axis is a specialized system in the brain that controls the production of cortisol as a hormonal mediator of a lot of physiological processes. Now the manuscript is done, submitted, so fingers crossed for a good outcome. And just a glimpse of the results there is that because ME patients, because of the symptoms, the core symptoms of ME patients can be linked to low cortisol, we were trying to understand the neurobiological basis of that. And for our surprise, when we go for the look in the tissue-based evidence of why the HPA axis is affected, we see almost a complete depletion of CRH neurons. So the magnitude of the changes in the brains of people affected by ME seem to be very, very much, very high, much more than we expected initially. That is also reflected in pituitary system. So here is gene expression profile of downstream genes related to CRH signaling. And we also detect lower cortisol. So basically, we actually are the first study to see the 3 levels of HPA axis in postmortem research and in context of ME as well. So we provided the first framework for neurobiological research in that. Well, so basically, this also means that low cortisol can be one of the explanations or partial explanations for a lot of hallmark symptoms of the disease. It's not one size fits all, but that's a working hypothesis of ours. Another thing that we are touching upon now, it is because CRH neurons innervate a lot of other hypothalamic systems, and we see that a lot of the patients that we recruited so far have sleep disturbances. We are working with orexin neurons, so basically they control sleep-awake cycles in the hypothalamus, and we are now doing staining in the brains of those people in the PVN cohorts, and the pilot study already showed changes as well, decrease of orexin neurons. The second work plan is basically the same. We leverage our expertise in histopathology and basically try to identify microglial changes as a driver of neuroinflammation, since neuroinflammation is deemed to be one of the mechanisms for ME/CFS pathophysiology. We have 2 work plans on that. One of them it is in situ characterization, but we also have fresh tissue that we dissociate in cell sorts, microglia, T and B cells. This is ongoing. I'll show some of the results for you. And this is also ongoing, but no analysis because we expect to only do them when we have 10 by 10 patients collected, 10 controls and 10 ME. It is interesting, something that also the in situ results show is that we do not see infiltration of T or B cells that is very different from the periphery. So that's something that we still have to make sense of. So we started to see the microglia cells there, and something that was like striking for us is that we do not observe more microglia, or we would expect as a neuroinflammatory condition there is infiltration of microglia or increase of microglia numbers. But rather than that, the microglia there, they are dystrophic. And I mean by that, they are fragmented, and the cell body is not connected to the processes, and the processes are also fragmented in itself. This is somewhat a new histopathological feature. Not a lot of people in the world had described that. Not a lot of conditions have that. There's no harmonized system to describe this. But we certainly do see that in the brains of ME patients. And something that is interesting is not only in the hypothalamus, but seems to be a widespread effect, both in gray and white matter. So in the patients that donate their brains, possibly end-stage patients, we do not see signs of classical neuroinflammation, but we see dystrophic, rather senescent microglia. Likewise, we see HLA is a proxy for microglia activation, and we only see indication of discrete increase in numbers of HLA. So again, we don't see robust signs of neuroinflammatory, as was our initial working hypothesis. And as I said, we do not see, or we do not have evidence so far of infiltration of T or B cells. One of the main mechanisms of action of microglia, or the main functions of a microglia, phagocytosis, and that is done through engulfing of particles in phagolysosomes that are characterized by CD68. So basically, we stain now CD68, and we also see a little bit of mixed results. So in the PVN, where we see loss of CRH neurons, we do see increase of CD68 positive microglia, which could be indication that there is a microenvironmental defect or microenvironmental injury. But in other parts of the brain, here bring the parietal cortex, we do not see that. So it seems to be triggered by local events rather than a widespread mechanism. We are following up on that, and more than these other markers of microglia activation. So we were very happy with our expertise in histopathology. We are continued that to also contemplate other cell types, and then we also felt the need that we should integrate other tools in our analysis, and that led to a second grant, that is brain multi-region single-cell transcriptomics of ME/CFS. Basically here, the plan is that we're going to have 3 brain regions, the hypothalamus, based on the findings that I showed to you, frontal cortex and hippocampus because of the cognitive problems that ME/CFS patients present. And we're going to use, in combination of histopathology, single-nuke RNA-seq, so detect genome-wide changes in the brains of ME/CFS. That we will understand why the underlying mechanisms of the changes that we observe at the cell level, and possibly have potential therapeutic targets. A second level of this grant, it is to try to harmonize in collaboration with NEWS the immune changes that we see in the brain and immune changes that we see in the periphery. So basically, we're going to compare data sets from the periphery, the blood, with the data sets from the microglia cells. And this will be done by Jennifer. She's a PhD student starting in February now, so the next meeting she will be here. And with these, I appreciate your attention, and if you have any questions or any other comments, happy to address them.
Any questions from the audience? How did you turn it on?
It's on.
Yeah.
Yeah. Okay. Yeah. For somebody who, you have to keep in mind that half of the audience doesn't know what HLA-DR is and what the parental cortex is.
Sure.
So, what would be the take-home message you think for patients? What is the key finding for you, and why is it important?
Sure. I think the key finding is that we do have microglia changes, as one would assume.
What are microglia?
Yeah, microglia. With HLA, it is present in microglia.
What are microglia?
Yeah, microglia are the immune cells in the brain, sorry. And there are changes, but that changes are not typically what we would expect or what has been speculated. So I think we have to reframe our thinking of what it is ME pathophysiology in the context of the brain.
So what have to be reframed? What view do we have to revise?
Yeah, well, we also have to put it in temporal mark, right? ME/CFS, it is thought to be a neuroinflammatory throughout the whole disorder, and might not be true. It might be that after a certain point, the neuroinflammation stops because the cells are just senescence. The cells cannot keep up with their continuous activation. So I would say that that's probably what these results indicate.
The nerve cells become exhausted.
Yeah, exactly.
Yeah, okay. Interesting. Thank you
Yeah,
very much.
thank
Very
you.
clear.
Maybe,
Yeah.
maybe related to that, what you said that the microglia becomes senescent. Do you think this is typical or atypical for a neuroinflammatory process, right? To put it a bit more in context, what you see.
Yeah, I think it is. We have two things here. We have severity of disease. So we have to admit that the donors are in the brain link. They probably are the more severe donors. And we have chronicity. So the combination of both lead to this phenotype that we see, probably.
Yeah, but would also mean, for example, in other, let's say in MS, or where you also see neuroinflammation, do you ultimately also end up with senescence with microglia or not? Or is this maybe something very unique for ME/CVS?
So it doesn't seem to be pathology specific.
Wait a second, Felipe.
Yeah.
Yeah. So it's not specific.
So yeah, no, it is something that we can have insights from other disorders. Some people have already reported in Alzheimer's disease. We start to see more in MS as well, in some types of lesions. So it is a very new pathological classification. There's no consensus on that, so we also have to be mindful of this.
I think it's so important also to mention that now the first time we look in the brains of people with ME/CVS, also with general pathological knowledge,
Yep.
the name myalgic muscles encephalomyelitis means inflammation of brain and spinal cord. Well,
Repeat the last sentence.
yeah. So the name myalgic encephalomyelitis means problems with the muscles, myalgic. Encephalomyelitis means that the brain, both brain and spinal cord are inflamed. And now we're so happy to have the brains to have a consistent look at it as also a general neuropathologist next to the hypothalamus and deep diving in the microglia, and we don't see this encephalomyelitis, no inflammation. These microglia cells are fragmented, dystrophic. We even don't agree on the name of these microglia, so we don't know what these look like. Senescence. We also see senescence in other diseases. This type of in dysmorphic microglia have been described in other diseases. So we dive deep into it now, genome-wide. And we will look also everywhere, but also at the hypothalamus, because our attention goes to the hormonal.
Neuroinflammation in the most classical sense because there's no TOB cell.
Yeah.
Yeah.
So we have to go to the next presentation, but you